So, if you break your arm, an X-ray gives you a pretty clean binary answer, right?
Oh, absolutely. It's either broken or it's not.
Exactly. The doctor just, points to that jagged white line, and there's really no debate.
Yeah, it's very straightforward.
But what if I told you that for decades, the absolute baseline map we've used to diagnose human liver disease has been, well, fundamentally wrong?
It's honestly wild to think about.
Right. So welcome to the deep dive. We are taking you with us today to relentlessly unpack this really groundbreaking April 2026 Nature paper.
And this paper, it finally, for the very first time, creates a true spatial atlas of a healthy human liver.
Which completely changes how we understand the organ.
It really does. Because up until this study, we were looking at diagnostic maps that were, frankly, heavily corrupted, and we didn't even fully realize it.
We were essentially flying blind. So for you listening, we are going to explore the literal geography of your liver today, down to the microscopic pixel.
And we'll see how human biology diverges wildly, like completely diverges from what we thought we knew based on animal models.
Yeah, and just a quick heads up for you, this deep dive is going to be incredibly dense. But, in the best way possible.
Tacked with aha moments.
Exactly. We are leaving no concept unexplained. So, to start, I want you to step into an operating room. Just, imagine a surgeon is removing a piece of a liver.
Okay, setting the scene.
Right. Historically, when scientists wanted to study a quote-unquote normal human liver just to establish a baseline, they basically pulled tissue from one of two sources.
Right, the traditional control.
Yeah. The first was neurologically deceased donors. So people who were brain dead but kept on life support.
Right.
And the second was what they call adjacent normal tissue, which is the visually healthy-looking liver flesh sitting right next to a surgically removed tumor.
And, both of those sound totally reasonable on the surface.
They really do. You'd think, okay, healthy tissue is healthy tissue.
But there is a massive hidden variable here. Let's look at the neurologically deceased donor livers first.
Okay, the NDDs.
Right. These patients are undergoing extreme systemic metabolic stress.
Yeah.
The body is on prolonged mechanical ventilation.
Which is not exactly a natural state.
Not at all. And it's receiving non-enteral nutrition, meaning, IV food rather than digesting normally through the gut.
Right, so the liver isn't even processing food the way it normally would.
Exactly. Plus, it's being pumped full of medications just to stabilize blood pressure. So all of that artificially alters the liver's genetic behavior before the tissue is even collected.
Wow, so the baseline is already skewed.
Heavily skewed. And then there's variable ischemia time.
Ischemia being when the tissue is starved of blood flow.
You got it. The longer that organ sits without blood flow, the more the RNA inside the cells just degrades unpredictably. You weren't getting a healthy map. You're getting like a map of a liver in crisis.
Okay, that makes complete sense. If a liver is essentially on life support, it's not acting normally.
Not even close.
But what about that second option? The adjacent normal tissue from a living patient who, just had a tumor removed.
Ah, yes. The adjacent normal.
Because if I pull tissue from the healthy margin, like a few centimeters away from the cancer, shouldn't that be a pristine control sample? It looks fine, right?
And you are hitting on the exact assumption the scientific community made for decades.
Wait, really?
Decades.
Decades. Because under a microscope, that adjacent tissue looks perfectly healthy. The cellular structure seems completely intact.
But looks can be deceiving.
Exactly. Because this paper looked at transcriptomics, which completely shattered that illusion.
Okay, let's unpack this for a second.
Yeah.
Transcriptomics. That's the study of the complete set of RNA transcripts produced by the genome, right?
Right. It doesn't just show us what the cell physically looks like. It shows us exactly which genetic blueprints are being actively read and translated into proteins at that exact second.
It's like looking at the factory floor instead of just the outside of the building. Perfect analogy. And when the researchers looked at the transcriptomics of that adjacent normal tissue, they realized it was profoundly compromised by the neighboring disease.
Wow. So it's like trying to judge the baseline everyday stress level of a city by only interviewing people who live right next door to a massive, deafening construction site.
That is a phenomenal way to frame it.
Right. They might look perfectly fine sitting calmly on their front porch, but their blood pressure is through the roof, and they haven't slept in weeks.
Yes, exactly. So to get around this, the researchers in this paper used tissue from live human donors, or LHDs.
Live human donors. Okay.
Right. These are highly screened, rigorously evaluated completely healthy people who are altruistically donating a piece of their liver to save a life.
Which is incredible in itself.
It really is. And this finally gave the team a true, uncorrupted baseline of human liver gene expression.
So when they finally compared these true, healthy, live donors to that adjacent normal construction site tissue, what was the actual difference at the molecular level?
The differences were staggering. I mean, let's look at the everyday canonical jobs of liver cells, which we call hepatocytes.
Okay, the hepatocytes.
In the adjacent tissue, the genes responsible for the urea cycle were drastically downregulated, especially an enzyme gene called ASS1.
Okay, so ASS1 is dropping, and the urea cycle, just to remind everyone, that's the heavy machinery your liver uses to convert highly toxic ammonia, which is a byproduct of protein metabolism, into urea, so you can safely pee it out.
Right, it is a critical daily chore.
Yeah.
But in the tumor-adjacent tissue, that machinery was just powering down.
Wait, so the garbage disposal was just turning off?
Basically, yeah.
But if those enzymes drop, the liver is essentially failing to filter ammonia properly. That sounds dangerous.
Well, it doesn't stop entirely, but it vastly reduces its capacity. The cells are essentially triaging their energy.
Oh, wow.
Yeah, and the researchers also saw significant drops in cytochrome P450 enzymes, specifically things like CYP2E1 and CYP2T8.
And those are your liver's primary detoxifiers, right? They break down everything from Tylenol to environmental toxins.
Exactly, downregulated. And they also saw a drop in PCK2, which is a key gene for gluconeogenesis.
Gluconeogenesis.
Yeah.
That's the really energy-intensive process of generating new glucose from scratch to keep your blood sugar stable.
Right. So the adjacent tissue was literally abandoning its primary biological jobs.
Because it was bracing for impact. I mean,
Yes.
It was too busy dealing with the tumor next door.
Precisely. And concurrently, that adjacent tissue showed a massive spike in immune-related genes,
Like what?
specifically calprotectin genes like
Wait.
S100A8 and S130A9.
S100A8 and S100A9. Okay, so you can think of calprotectin proteins as like cellular fire alarms.
That's a great way to think about it. When they spike, they trigger widespread inflammation, signaling to the immune system that the tissue is under threat.
Right.
And the live donor livers, they didn't have these alarms blaring at all. They were just peacefully doing their metabolic jobs.
Man. So if I'm synthesizing this correctly for you listening, just to do a quick bullet point recap.
Yeah, let's summarize.
First, live human donors are the only true baseline. Second, adjacent tissue is actually metabolically suppressed and highly immune activated. So for decades, medical researchers thought they were looking at a normal baseline, but they were actually looking at a liver that had abandoned its day job to fight a local war.
It's completely wild.
It completely changes how we should read past medical studies. So here's a question for you listening. Think about other medical research you've read. How often is the control group secretly compromised like this?
It definitely forces us to question a massive amount of historical literature. But, establishing this new uncorrupted baseline was really only the first step.
Right, because now they had to actually map it.
Exactly. To truly understand how this healthy liver operates, the researchers had to map its geography. Because the liver isn't just, it's not a homogeneous mush of identical cells.
No, it has a highly specific architecture that dictates how it functions. And this is where we welcome you to the lobule.
Yes, the lobule.
I want you to visualize this. If you look at a liver under a microscope, it's organized into millions of tiny, repeating hexagonal structures.
Like a honeycomb.
Exactly. These are the lobules. And each hexagon is minuscule, only about 0.5 millimeter to 1 millimeter in diameter.
And the architecture here is at everything. Blood flows into these hexagonal lobules from the outside edges, which we call the portal veins.
Okay, outside edges are portal.
Right. The blood then washes over the liver cells, flowing inward toward the center of the hexagon, and it eventually drains out through the central vein.
So think of it like a spatial river system.
Sure.
The portal zone on the outside edges is like the rushing rapids where fresh water enters.
Right. Highly oxygenated.
Packed with fresh nutrients straight from the gut.
Ah.
And as the blood flows inward, the liver cells along the path greedily consume that oxygen and those
They
nutrients.
just gobble it up.
So by the time the blood reaches the central zone, the very middle of the hexagon, it's like a deep palm lake where the water pools. The oxygen is heavily depleted.
And that physical gradient of oxygen and nutrients creates a phenomenon called zonation.
Zonation, okay.
Yeah. Hepatocytes perform entirely different jobs depending on where they live along that river.
Oh, that makes sense.
A cell living in the oxygen-rich portal rapids behaves fundamentally differently than a cell in the oxygen-depleted central lake. And to map this zonation, the researchers deployed this massive arsenal of spatial transcriptomic technologies.
And spatial transcriptomics is just wild because it doesn't just tell you what RNA is present, it tells you exactly where it came from geographically.
Exactly. They used a technology called 10x Visium.
10x Visium, how does that work?
It essentially lays the liver tissue over a microscopic grid of barcoded spots. So when the tissue releases its RNA, the grid captures it and tags it with a barcode based on its precise physical location.
That is so cool.
Right. And they also used single nucleus RNA sequencing, or snRNA-seq, to get really granular data. Plus, something called MERFISH.
MERFISH. What is MERFISH?
It acts like a molecular GPS system. It basically tags specific RNA molecules with glowing fluorescent colors, so you can literally see where certain genes are turned on under a microscope.
So they basically built Google Maps for the liver.
They really did. But, to orient a map, you needed a landmark.
Right. If I drop you in the middle of a city, you look for a famous skyscraper to know which way is north.
Exactly. And the primary landmark they used was that specific detoxification gene we mentioned earlier, CYP2E1.
Okay.
In humans, this gene is heavily pericentral. It almost exclusively lives near that calm, low-oxygen lake in the center of the lobule.
So it's like the central beacon.
Right. By locating the intense glowing signal of that specific gene, the computer algorithms could instantly orient themselves. They can say, okay, we're in the central zone, and map the relative location of every other cell from there.
Amazing. Okay, let's do another quick recap. Lobules are tiny hexagons. Portal equals high oxygen entry on the outside. Central equals low oxygen exit on the inside. And cell functions change based on their location.
Spot on.
So here's a question for you listening. If you were a cell, would you want to live in the high-energy portal zone, catching all that fresh oxygen, or the specialized central zone?
Well, you would probably assume you'd want the portal zone, right? Especially if you looked at a mouse liver.
Okay, here's where it gets really interesting, because this brings us to the most profound evolutionary revelation in the paper. The researchers didn't just map humans, they mapped mice, wild boars, domestic pigs, and cows to see how our geography compares.
And this is where my mind was completely blown.
Mine too. Because in a mouse, the heavy metabolic lifting, the really energy-intensive jobs, that all happens in the portal zone, right where all the oxygen is.
Which makes perfect mechanical sense. You build your biggest factories right next to the power plant.
Right. But the human liver pushes its major metabolic functions to the pericentral zone.
It is a massive, unprecedented evolutionary shift. In humans, the entire heavy machinery for the urea cycle, specifically genes like ASL, AGS, and OTC.
With ASL, NAGs, and OTC.
Yes, those are the genes responsible for clearing toxic ammonia. They are physically packed up and moved to the center in humans.
That's crazy.
And it gets weirder. The machinery for gluconeogenesis, making new sugar from scratch, like that PCK2 gene, is shifted to the center. Even the glucose transporter GLUT2, which is coded by the SLC2A2 gene envelope.
Wait, SLC2A2 is the gene for the GLUT2 transporter.
Exactly. Also pericentral, even a master transcription factor called HNF4A, which is like a boss gene that commands other liver genes what to do. It's completely pericentral in humans, whereas it sits in the portal zone in mice.
Okay, wait. I have to challenge this because it feels like a biological paradox.
Go for it.
If the central zone has the absolute least oxygen in the entire lobule, why on earth is the human body forcing the heaviest, most energy-demanding metabolic lifting down there? That seems completely counterintuitive. The cells must be constantly starved for energy while doing the hardest work.
You're touching on the exact friction the scientists explored. It seems completely backward, but it points to something called the glucose paradox.
The glucose paradox. Unpack that for us.
So, you see, the liver job is to store sugar as glycogen. But it replenishes those stores using different chemical pathways depending on the zone.
Okay.
The pericentral hepatocytes, the ones in that low oxygen center, have the unique ability to absorb glucose directly from the bloodstream to make glycogen. But the periportal cells, out in high oxygen rapids, they actually prefer to use lactate to build their glycogen.
Oh. So because human brains are so massive, and our brains demand such an absurd, constant supply of direct glucose compared to a mouse or a cow,
Right.
Our livers had to essentially rewire their entire geography. We heavily armed that central zone to manage direct glucose metabolism, even if it meant starving those cells of oxygen.
It is a profound adaptation to feed the human brain. We completely reorganized our liver's entire metabolic factory. And they also found these unique lobule genes in humans.
Oh, midlobule, like halfway between portal and central.
Exactly. Genes like HST17B13 and STC1 that just don't follow the same patterns in other mammals.
Wow. Okay, quick recap. Humans are entirely unique. We shifted massive metabolic jobs to the low oxygen center, unlike mice, pigs, or cows.
Yes, we are the outliers.
So question for you listening. Why do you think humans evolved this unique central heavy metabolic factory compared to a pig or a cow? I mean, beyond just the brain, what else is driving that?
It's a great question to ponder, but, it's not just the hepatocytes doing all this work. The liver is a bustling city. You need more than just office workers. You need infrastructure.
Right, which brings us to the cellular chat, the ecosystem of the liver, the non-parenchymal cells, or NPCs.
Exactly, the supporting cast. The spatial mapping showed that the liver relies heavily on three main NPCs. First, you have the liver sinusoidal endothelial cells, or LSECs.
And you can think of LSECs as incredibly porous pipes lining the blood vessels, letting nutrients slip through.
Perfect. Second, you have the Kupffer cells.
Kupffer cells, those are the liver's resident macrophages, right? The immune bouncers that eat bacteria.
Exactly. And third, you have the fibroblasts, which are basically the structural engineers laying down the scaffolding.
Okay, so LSECs, Kupffer cells, and fibroblasts.
Right. And what's brilliant about the transcriptomics here is that it allowed researchers to see how these cells actually communicate.
They're constantly texting each other across the zones.
Yes. They use chemical signals called ligands, which are text messages, and they have receptors, which act like the cell phones receiving the message.
Okay, take a second to explain the actual biological mechanism there. When we say they quote-unquote text each other, what actually happens physically inside the cell?
Okay, let's look at the central zone. The endothelial pipes, the LSECs, secrete a specific chemical text message, a ligand, into the surrounding fluid.
A ligand called WNT2, for example.
Exactly, WNT2. That chemical floats over and physically docks into a receptor, specifically the FZD5 receptor on the surface of a nearby hepatocyte.
WNT2 docks into FZD5.
Right. And when that chemical docks, it's like a key turning in an ignition. It triggers a physical chain reaction inside the hepatocyte that actually unspools specific sections of its DNA, turning on those heavy-duty central zone genes.
Wow.
Yeah. And the fibroblasts do this too, sending signals like BMP9 and PMP10. The supporting cells are literally commanding the hepatocytes on how to behave based on their physical location.
That's just incredible. But the detail about the NPCs that really caught my attention was the Kupffer cells, those immune bouncers.
Ah, yes. The Kupffer cell distribution is another wild departure from mice.
Tell them.
In mice, the Kupffer cells are clustered out in the portal zone, the rapids, which makes intuitive sense for immune defense.
Right. They're standing right at the front door, catching gut bacteria the exact second it enters the liver.
But in humans, the Kupffer cells are heavily shifted away from the front door. They're clustered in the pericentral zone.
Which is just, why put your security guards at the back door? It feels like we traded front door security for something else entirely.
We did.
Right.
And what's fascinating here is why. It comes right back to our unique, heavy-lifting human metabolism.
Because of the glucose paradox stuff.
Yes. Because we force our pericentral hepatocytes to do such intense metabolic labor in a really low oxygen environment, those central cells suffer massive wear and tear.
They're basically burning out.
Exactly. They die at a much higher rate. So we likely need the Kupffer cells stationed in the center to quickly clean up the microscopic debris of those overworked, dying hepatocytes.
So they're acting more like garbage men than bouncers in this context.
Exactly. If they don't clean it up instantly, the dead cells would cause cascading, catastrophic inflammation.
Oh, man. So quick recap. NPCs text each other across zones using things like WNT2 and BMP9. And human Kupffer cells guard the center, acting as garbage men, unlike mice.
Right.
So here's a question for you listening. If the Kupffer cells are clustered in the center, what does that mean for how human livers actually fight off infections entering from the portal vein?
It's a huge vulnerability.
It really is. We mapped our immune cells to protect us from our own metabolic exhaust, rather than outside invaders.
And that sheer biological compromise, that vulnerability, perfectly sets up the final major discovery of this paper.
The sneak attack of steatosis, or what we commonly call fatty liver disease.
Right. Now, because the live human donors used in this study were so robust and healthy overall, their livers could be used for transplant even if they had up to 20% fat accumulation.
20% fat is still considered healthy.
In a transplant context, yes. And this gave the scientists an accidental but totally perfect map of early steatosis.
The absolute genesis of fatty liver disease before it turns into a chronic pathology like MASLD.
Exactly. And to figure this out, they used an incredibly clever AI algorithm. They fed the traditional stained H&E microscope images into a machine learning program.
H&E imaging pixels.
Right. And the AI could actually analyze those pixels to quantify the exact size and density of microscopic lipid droplets, literally bubbles of fat inside individual cells.
And then they overlaid that fat map onto their spatial gene expression map.
Yes. And they discovered that fat doesn't just accumulate uniformly. It has a geographical preference.
Let me guess, it starts in the center.
It starts centrally. Steatosis strongly prefers to start in that highly stressed pericentral zone.
Of course it does.
But it was the genetic reaction to this fat that was the true revelation. Inside your cells, you have two separate power systems.
Okay, walk us through the power systems.
You have your main nuclear DNA, which creates the primary proteins for your mitochondria, the cell's power plants.
You're right.
But the mitochondria also have their own tiny circular loops of local DNA.
Okay, so a main grid and a local grid.
Exactly. And as those fat droplets accumulated in the central zone, the scientists saw a sharp decline in the mitochondrial proteins being coded by the cell's main nucleus.
Oh, wow. Think about the biological friction here. You have lipid droplets literally swelling up, physically crowding the cellular machinery.
Yes. The main nuclear power grid is physically and metabolically choked off by the accumulating lipids.
So the cell senses this catastrophic energy failure.
Yeah.
And it panics.
It absolutely panics. It triggers a localized survival mechanism. The tiny local loops of mitochondrial DNA kick into absolute overdrive.
They ramp up their own transcripts.
Right, rapidly increasing to try and keep the power plants running and burn off the invading fat.
So basically, what does this all mean? It's like the main power grid failing, so the local backup generators kick into overdrive just to keep the lights on.
That is the perfect analogy. That localized shift from nuclear to mitochondrial control is the earliest molecular whisper of the disease.
Man. Okay, final recap for this section. Fat accumulation starts in the center, causing the cell's power grid to switch from nuclear to local mitochondrial control.
Yes.
So question for you listening. Could this early power switch be the actual key to reversing fatty liver before it becomes a full-blown disease?
Well, because it's a localized compensatory survival mechanism, and because we can now see this spatially, at the very earliest stages, it absolutely opens up a whole new world for intercepting the disease.
Right. Because as we zoom out to the big picture here, you cannot fix a broken engine if you don't have a blueprint of what a working engine actually looks like.
Exactly. For decades, we have been trying to cure human liver diseases by looking at the blueprints of stressed-out tumor-adjacent livers.
Livers in crisis.
Right? Or we've been using the blueprints of mice, which we now know are fundamentally geographically reversed compared to us.
It's wild to think about the wasted effort. But by establishing this true, spatially resolved baseline of a healthy, live human donor, we finally have the correct map.
We can see exactly where the cellular text messages are supposed to be sent.
Exactly where the glucose is being processed.
And exactly how the backup generators fail when fat moves in. It's a monumental leap forward.
It really is. And, that leads me to a final, slightly provocative thought for you to mull over as we wrap up today.
Oh, let's hear it.
If our liver zonation is so wildly different from a mouse's, how many decades of liver disease drugs failed in human trials simply because we were trying to fix a mouse map inside a human body?
It's a sobering thought. Thousands of drugs, probably.
Right. And on the flip side, if we now know exactly how a healthy human liver organizes itself down to the exact pixel, down to the exact zonation of immune cells and metabolic factories, what does this mean for the future of lab-grown organs?
Oh, like 3D printing.
Exactly. Could we one day use this exact biological blueprint to 3D print a fully functional human liver, complete with its own central heavy metabolic zoning, and just entirely eliminate the need for transplants?
Having this precise spatial instruction manual makes that science fiction concept significantly closer to reality. We actually have the code now.
We have the code. Well, thank you for joining us on this incredibly deep molecular journey today. We covered a lot of ground, from the rushing portal rapids to the quiet, overworked central lakes of your liver lobules.
It was a dense one, but so important.
Absolutely. So keep questioning the baselines and everything you learn, because sometimes what we blindly accept as the control group is actually the biggest variable of all. Catch you on the next deep dive.